Good morning, everybody. Welcome to the subject of biotechnology. Today we deal with the fundamentals of molecular biotechnology on behalf of New Age Cosmetic Wide EdTech Healthcare Consultancy. I welcome all of you to my daily lectures. Excuse me. Now, starting with the subject of the fundamentals of molecular biotechnology, here are the portions to be covered. The development of molecular biotechnology, DNA, RNA, and protein synthesis, recombinant DNA technology, chemical synthesis, amplification, sequencing of DNA, bioinformatics, genomics and proteomics, manipulation of gene expression in prokaryotes, heterologous protein production, eukaryote cells, directed mutagenesis, and protein engineering. There could be reference in other chapters also that we talked, but this, as per the syllabus, we need additional coverage. So here we proceed. Molecular biotechnology is an exciting scientific discipline that is based on the ability of the researchers to transfer specific units of genetic information from one organism to another. This conveyance of a gene or genes relies on the techniques of genetic engineering, recombinant DNA technology. The objective of recombinant DNA technology is often to create a useful product or a commercial process. Dear students, this is in the best interest of those students who do not have access to good, sound learning material, study material. We give credits to all the authors of the various textbooks that we are referring to. Please make necessary notes. Do not commercialize anything. Use it to the best of your study purposes to achieve your goals of passing the various qualifiers, like the NEET biology, NEET exams, all the Institute of Medical Sciences, Council of Scientific and Industrial Research, National Eligibility Tests, and similar tests. Okay, proceeding. Essential molecular biotechnology laboratory techniques, including chemical synthesis of genes, the polymerase chain reaction, PCR, and DNA sequencing, are discussed. Developments in sequencing technologies have led to the sequencing of the entire genomes of many organisms, and this has enabled researchers to begin to understand organisms from their sequences and to identify novel genes with potentially useful functions. In addition to isolation, cloning of genes, it is important that these genes function properly in a host organism. To this end, strategies for optimizing the expression of a cloned gene in either prokaryotic or eukaryotic cells are reviewed. Finally, procedures for modifying cloned genes by the introduction of specific nucleotide changes and in vitro mutagenesis to enhance the properties of the target proteins are examined. Together, the chapters in Part 1 provide conceptual and technical underpinnings for understanding the applications of molecular biotechnology that are described in the ensuing chapters. The emergence of molecular biotechnology long before we knew that microorganisms existed, or that genes were the units of inheritance, humans looked to the natural world to develop methods to increase food production, preserve food, and heal the sick. Our ancestors discovered that grains could be preserved through fermentation into beer, that storing horse saddles in the warm, damp corner of the stable resulted in the growth of a saddle mold that could heal infected saddle sores, and that intentional exposure to a contagion could somehow provide protection from an infectious disease and subsequent exposure. Since the discovery of the microscopic world in the 17th century, microorganisms have been employed in the development of numerous useful processes and products. Many of these are found in our households and backyards. Lactic acid bacteria are used to prepare yogurt and probiotics, insecticide producing bacteria, and sprayed on many of the plants from which the vegetables in our refrigerator were harvested. Nitrogen-fixing bacteria are added to the soil used for cultivation of legumes. The enzymatic stain removers in laundry detergent came from microorganisms, and antibiotics derived from common soil microbes are used to treat infectious disease. These are just a few examples of traditional biotechnologies that have improved our lives up to the early 1970s. However, traditional biotechnology was not a well-recognized scientific discipline, And research in this area was centered in departments of chemical engineering and occasionally in specialized microbiology programs. In a broad sense, biotechnology is concerned with the production of commercial products generated by the metabolic action of microorganisms. More formally, biotechnology may be defined as the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. The term biotechnology was first used in 1917 by a Hungarian engineer called Erecki to describe an integrated process for the large-scale production of pigs by using sugar beets as a source of food. According to Erecki, biotechnology was all lines of work by which products are produced from raw materials with the aid of living things. This fairly precise definition was more or less ignored. For a number of years, the term biotechnology was used to describe two very different engineering disciplines. On the one hand, it referred to industrial fermentation, on the other, it was for the study of efficiency in the workplace, what is now called ergonomics. So here you see raw material, upstream processing, fermentation, biotransformation, downstream processing, pure product. But on the other hand, it was used for the study of efficiency in the workplace, which is now called ergonomics. This ambiguity ended in 1961 when the Swedish microbiologist Carl Gran Hedén recommended that the title of a scientific journal dedicated to publishing research in the fields of journal applied microbiology and industrial fermentation be changed from the Journal of Microbiological and Biochemical Engineering and Technology to Biotechnology and Bioengineering. From that time on, biotechnology has clearly and irrevocably been associated with the study of the industrial production of goods and services by processes using biological organisms, systems, and processes. It has been firmly grounded in expertise in microbiology, biochemistry, and biochemical engineering. An industrial biotechnology process that uses microorganisms for producing a commercial product typically has three key stages: upstream processing, preparation of the microorganisms and the raw materials required for the microorganisms to grow and produce the desired product; fermentation and transformation growth, fermentation of the target microorganisms in a large bioreactor, usually greater than 100 liters, with the consequent production biotransformation of a desired compound, which can be, for example, an antibiotic, an amino acid, or a protein. Downstream processing, purification of the desired compound from either the cell medium or the cell mass. Biotechnology research is dedicated to maximizing the overall efficiency of each of these steps and to finding microorganisms that make products that are useful in the preparation of foods, food supplements, and drugs. During the 1960s and 1970s, this research focused on upstream processing, bioreactor design, and downstream processing. These studies led to enhanced bioinstrumentation for monitoring and controlling the fermentation process and to efficient large-scale growth facilities that increase the yields of various products. The biotransformation component of the overall process was the most difficult phase to manipulate. Commodity production by naturally occurring microbial strains on a large scale was often considerably less than optimal. Initial efforts to enhance product yields focused on creating variant mutants by using chemical mutagens, ultraviolet radiation to induce changes in the genetic constitution of existing strains. However, the level of improvement that could be achieved in this way was usually limited biologically. If a mutated strain, for example, synthesized too much of a compound, other metabolic functions often were impaired, thereby causing the strain's growth during large-scale fermentation to be less than desired. Despite this constraint, the traditionally induced mutagenesis and selection strategies of strain improvement were extremely successful for a number of processes, such as the production of antibiotics. The traditional genetic improvement regimens were tedious, time-consuming, and costly because of the large numbers of colonies that had to be selected, screened, and tested. Moreover, the best result that could be expected with this approach was the improvement of an existing inherited property of a strain rather than the expansion of its genetic capabilities. an industrial biotechnology process that uses microorganisms for producing a commercial product typically has three key stages. Upstream processing, preparing of the microorganisms and the raw materials required for the microorganism to grow and produce the desired product. Fermentation and transformation, growth fermentation of the target microorganisms in a large bioreactor, usually greater than 100 liters, with the consequent production biotransformation of a desired compound, which can be, for example, an antibiotic, an amino acid, or a protein. 3. Downstream processing, purification of the desired compound from either the cell medium or the cell mass. Biotechnology research is dedicated to maximizing the overall efficiency of each of these steps and to finding microorganisms that make products that are useful in the preparation of foods, food supplements, and drugs. During the 1960s and 1970s, this research focused on upstream processing, bioreactor design, and downstream processing. These studies led to enhanced bioinstrumentation for monitoring and controlling the fermentation process and to efficient large-scale growth facilities that increase the yields of various products. The biotransformation component of the overall process was the most difficult phase to manipulate. Commodity production by naturally occurring microbial strains on a large scale was often considerably less than optimal. Initial efforts to enhance product yields focused on creating variants, mutants, by using chemical mutagens or ultraviolet radiation to induce changes in the genetic constitution of existing strains. However, the level of improvement that could be achieved in this way is usually limited biologically. If a mutated strain, for example, synthesized too much of a compound, other metabolic functions often were impaired, thereby causing the strain's growth during large-scale fermentation to be less than desired. Despite this constraint, the traditionally induced mutagenesis, and selection strategies of strain improvement were extremely successful for a number of processes such as the production of antibiotics. The traditional genetic improvement regimens were tedious, time-consuming, and costly because of the large numbers of colonies that had to be selected, screened, and tested. Moreover, the best result that could be expected with this approach was the improvement of an existing inherited property of a strain rather than the expansion of its genetic capabilities. Despite their limitations, by the late 70s, effective processes for the mass production of a wide range of commercial products had been perfected. Excuse me. Today, we have acquired sufficient knowledge of the biochemistry, genetics, and molecular biology of microorganisms to accelerate the development of useful and improved biological products and processes, and to create new products that would not otherwise occur. Distinct from traditional biotechnology, the modern methods require knowledge of and manipulation of genes, the functional units of inheritance, and the discipline that is concerned with the manipulation of genes for the purpose of producing useful goods and services using living organisms is known as molecular biotechnology. The pivotal development that enabled this technology was the establishment of techniques to isolate genes and to transfer them from one organism to another. This technology is known as recombinant deoxyribonucleic acid technology, DNA technology. Recombinant deoxyribonucleic acid, DNA technology. So that is recombinant DNA technology. And it began as a lunchtime conversation between two scientists working in different fields who met at the Scientific Conference in 1973. In his laboratory at Stanford University in California, Stanley Cohen had been developing methods to transfer plasmids called circular DNA molecules into bacterial cells. Meanwhile, Herbert Boyer of the University of California at San Francisco was working with enzymes that cut DNA at specific nucleotide sequences. Over lunch at a scientific meeting, they reasoned that Boyer's enzyme could be used to splice a specific segment of DNA into a plasmid, and then the recombinant plasmid that could be introduced into a host bacterium using Cohen's method. Let me repeat that. Over lunch at a specific meeting, they reasoned that Boyer's enzyme could be used to splice a specific segment of DNA into a plasmid, and then the recombinant plasmid could be introduced into a host bacterium using Cohen's method. recombinant DNA technology. It was clear to Cohen and Boyer and others that the recombinant DNA technology had far-reaching possibilities, as Cohen noted at that time. It may be possible to reintroduce in E. coli genes specifically specifying metabolic or synthetic functions such as photosynthesis or antibiotic production, indigenous to other biological classes. The first commercial product produced using recombinant DNA technology and was human insulin, which is used in the treatment of diabetes. The DNA sequence that encodes human insulin was synthesized, a remarkable feat in itself at that time, and was transplanted into a plasmid that could be maintained in this common bacterium, Escherichia coli. The bacterium host cells acted as biological factories for the production of the two peptide chains of human insulin, which after being combined could be purified and used to treat diabetes, or diabetics who were allergic to the commercially available porcine pig insulin. In the previous decade, this achievement would have seemed absolutely impossible. By today's standards, however, this type of genetic engineering is considered commonplace. By today's standards, however, this type of genetic engineering is considered commonplace. The nature of biotechnology was changed forever by the development of recombinant DNA technology. With these techniques, the maximization of the biotransformation phase of a biotechnology process was achieved more directly. Genetic engineering provided the means to create rather than merely isolate highly productive strains. Not long after the production of the first commercial preparation of recombinant human insulin, bacteria and then eukaryotic cells were used for the production of insulin, interferon, growth hormone, viral antigens, and a variety of other therapeutic proteins. Recombinant DNA technology could also be used to facilitate the biological production of large amounts of useful, low molecular weight biological compounds and macromolecules that occur naturally in minuscule quantities. Plants and animals became targets to act as natural bioreactors, producing new or altered gene products that could never have been created either by mutagenesis and selected or by crossbreeding. Molecular biotechnology has become the standard method for developing living systems, with novel functions and capabilities for the synthesis of important commercial products. Most new scientific disciplines do not arise entirely on their own. They are often formed by the amalgamation of knowledge from different areas of research. For molecular biotechnology, the biotechnology component was perfected by industrial microbiologists and chemical engineers, whereas the recombinant DNA technology portion owes much to discoveries in molecular biology, bacterial genetics, and nucleic acid entomology. In a broad sense, molecular biotechnology draws on knowledge from a diverse set of fundamental scientific disciplines to create commercial products that are useful in a wide range of applications. The Cohen and Boyer strategy for gene cloning was an experiment heard around the world. Once the concept was made public, many other researchers immediately appreciated the power of being able to clone genes. Consequently, scientists created a large variety of experimental protocols that made identifying, isolating, characterizing, and utilizing genes more efficient and relatively easy. These technological developments have had an enormous impact in generating new knowledge in practically all biological disciplines, including animal behavior, developmental biology, molecular evolution, cell biology, and human genetics. Indeed, the emergence of the field of genomics was dependent on the ability to clone large fragments of DNA into plasmids in preparation for sequence determination. Commercialization of molecular biotechnology. The potential of recombinant DNA technology reached the public with a frenzy of excitement, and many people became rich on its promise. Indeed, producing new or altered gene products that could never have been created either by mutagenesis and selection or by crossbreeding. Molecular biotechnology has become the standard method for developing living systems with novel functions and capabilities for the synthesis of important commercial products. Most new scientific products and do not arise entirely on their own. They are often formed by the amalgamation of knowledge from different areas of research. For molecular biotechnology, the biotechnology component was perfected by industrial microbiologists and chemical engineers, whereas the recombinant DNA technology portion owes much to discoveries in molecular biology, bacterial genetics, and nucleic acid enzymology. In a broad sense, molecular biotechnology draws on knowledge from a diverse set of fundamental scientific disciplines to create commercial products that are useful in a wide range of applications. The Cohen and Boy strategy for gene cloning was an experiment heard around the world, and I think we've explained that portion. And now the commercialization of molecular biotechnology. The potential of recombinant DNA technology reached the public with a frenzy of excitement. Many became rich on its promise. This figure down is schematic shows many scientific disciplines contributing to molecular biotechnology, which generates a wide range of commercial products. Molecular biology, microbiology, biochemistry, immunology, genetics, genetic, chemical engineering, cell biology, leading to molecular biotechnology. Effective in crops, drugs, vaccines, diagnostics, and livestock. Okay, and now we have a history of the entire of the various patents and genomic sequences and developments. In 1988, a US patent is granted for a genetically engineered mouse susceptible to cancer. In 1988, the PCR method is published. 1990, approval is granted in the United States for a trial of human somatic cell gene therapy, and the same year, Human Genome Project is officially initiated, followed by recombinant chyme machine is being used for cheese making in the United States. 1994 to 95, were detailed genetic and physical maps, of human chromosomes were published. 94, FDA announces that genetically engineered tomatoes are as safe as conventionally bred tomatoes. 1995, first genome sequence of a cell organism, the bacterium Haemophilus influenza, is completed. 1996, first recombinant protein erythropoietin exceeds $1 billion in annual sales. Next year, same year, 1996, complete DNA sequence of all the chromosomes of the eukaryotic organisms, the yeast Saccharomyces, is determined. And the same year, commercial planting of genetically modified crops begins. 97, cloning of a mammal, a sheep with differentiated cell nucleus, is accomplished. And 98, FDA approves first antisense drug. 99, FDA approves recombinant fusion proteins, diphtheria toxin, interleukin-2 for cutaneous T-cell lymphoma, I guess. Yes. 2000, Arabidopsis genome is sequenced, followed by monoclonal antibodies exceeding $2 billion annual sales, followed by development of golden rice, pro-vitamin A producing rice is announced, followed by over $33 billion being invested in US biotechnology companies. In 2001, human genome sequence. 2002, complete human gene microarrays, gene chips become commercially available. 2002, same year, FDA approves first nucleic acid test system to screen whole blood from donors for HIV and HCV. 2004, large-scale sequencing of the Sargasso Sea metagenome begins. 2005, NCBI announces that there are 100 gigabases of nucleotides. In 2006, two recombinant cancer vaccine becomes available to protect against cervical cancer. 2008, 2 billionth acre of genetically engineered crops is planted. 2009, FDA approves first drug produced in a genetically engineered animal, a goat. And indeed, within 20 minutes of the start of the trade on the New York Stock Exchange on 14th October 1990, the price of shares in Genentech, company founded by Cohen and Boyer with chemist and entrepreneur Robert Swanson, that produced recombinant human insulin, went from $35 to $89. This was the fastest increase in the value of any initial public offering in the history of the market. It was predicted that some genetically engineered microorganisms would replace chemical fertilizers and others would eat up oil spills. Plants with inherent resistance to a variety of pests and exceptional nutritional content would be created, and livestock would have faster growing times, more efficient feed utilization, and meat with low fat content. Many were convinced that as long as a biological characteristic was genetically determined by one or a few genes, organisms with novel genetic constitutions could be readily created. Today we see that despite the commercial hype that dominated reality at the beginning, this infatuation with recombinant DNA technology was not totally unfounded. A number of the more sensible versions of the initial claims, although trimmed in scope, have become realities. In the 25 years since the commercial production of recombinant human insulin, more than 200 new drugs produced by recombinant DNA technology have been used to treat over 300 million people with diseases such as cancer, multiple sclerosis, cystic fibrosis, and strokes, and to provide protection against infectious diseases. Over 400 new drugs are in the process of being tested in the human trials to treat Alzheimer's disease and heart disease, to name only two. Similarly, many new molecular biotechnology products for enhancing crop and livestock yields, decreasing pesticide use, and improving industrial processes such as the manufacture of pulp and paper, food, energy, and textiles have been created and are marketed. The impact on agriculture has been tremendous. According to the Food and Agriculture Organization of the United Nations, yield improvements of all major crops have decreased due to poor agricultural management practices, decreased acreage of arable land, and increased reliance on fertilizers and pesticides that diminish soil quality. To produce more food on less land, 13 million farmers in 25 countries are now planting genetically engineered crops on 300 million acres of land. These crops are predominantly corn, cotton, canola, and soybeans that are resistant to herbicides and insects. Over the last 40 years in the United States, genetically engineered crops contributed to $44 million in economic gains due to increased yields and lower production costs. The global market value of genetically modified crop is currently $7.5 billion. Small resource poor farmers among the beneficiaries of agricultural biotechnology. In a comparative study of small cotton farms in South Africa, it was found that the yield of cotton from plants that were genetically engineered to produce a bacterial insecticide was on average about 70% greater than those from non-genetically modified plants over 3 seasons. Higher yields and reduced pesticide and labor costs translated into double revenues despite the slightly higher cost of the transgenic seeds. Similarly, in India, farmers who planted genetically modified cotton increased their yields by 31% to 2008 while decreasing insecticide use of 39%. This resulted in an 88% increase in all profits for small farmers. The ultimate objective of all biotechnology research is the development of commercial products. Consequently, the molecular biotechnology is driven to a great extent by economics. Not only does financial investment currently sustain molecular biotechnology, but clearly the expectation of with financial gain was responsible for the considerable interest and excitement during the initial stages of its development. By nightfall on 14 October 1980, the principal shareholders of Genentech stock were worth millions of dollars. The unprecedented enthusiastic public response to Genentech encouraged others to follow. Between 1980 and 1983, about 200 small biotechnology companies were founded in the United States with the help of tax incentives and funding them from both the stock market, speculation, and private investment, like Herbert Boyer, who was first a research scientist at the University of California at San Francisco, and then a vice president of Genentech. University professors started many of the early companies. Much of the commercial development of molecular biotechnology has been centered in the United States. By 1985, there were over 400 biotechnology companies, including many with names that contain variants of the word gene to emphasize their expertise in gene cloning. Excuse me. Biogen, Amgen, Calgen, Angenix, Genix, and CanGene. Today there are about 1,500 biotechnology companies in the United States, 3,000 in Europe, and more than 8,000 worldwide, most in the health care sector. All large multinational chemical and pharmaceutical companies, including Monsanto, DuPont, Pfizer, Eli Lilly, Smith, GlaxoSmithKline Beacon, Merck, Novartis, and Hoffmann La Roche, to name but a few, have made significant research commitments to molecular biotechnology. During the rapid proliferation of the biotechnology business in the 1980s, small companies were absorbed by larger ones. Strategic mergers took place and joint ventures were undertaken. For example, in 1991, 60% of Genentech was sold to Hoffmann La Roche for $2.1 billion. Also, inevitably, for various reasons, there were a number of bankruptcies. This state of flux is a characteristic feature of the biotechnological industry. So, currently, the roster of biotechnology companies is extensive and includes those focused on cardiovascular disorders, tissue engineering, cell replacement, drug delivery, vaccines, gene therapy, antisense drugs, microarray detection systems, diagnostics, genomics, proteomics, and agricultural biotechnology. Concerns and consequences. While many people appreciate the potential of molecular biology, biotechnology, to solve important problems in agriculture, medicine, and industry, they recognize the need to be cautious about its widespread application. Indeed, one of the first scientific responses to this new technology was a voluntary moratorium on certain experiments that were thought to be potentially hazardous. This research ban was self-imposed by a group of molecular biologists, including Cohen and Boyer. They were concerned that combining genes from two different organisms might accidentally create a novel organism with undesirable and dangerous properties. Within a few years. However, these apprehensions were allayed as scientists gained laboratory experience with this technology and safety guidelines were formulated for recombinant DNA research. The temporary cessation of some recombinant DNA research projects did not dampen the enthusiasm for genetic engineering. In fact, the new technology continued to receive unprecedented attention from both the public and scientific community. Molecular biology, biotechnology, can contribute benefits to humanity. It can provide a wide range of infectious and genetic disease, providing opportunities to accurately diagnose, prevent, or cure a wide range of infectious and genetic diseases. Let me read that again. Molecular biotechnology can contribute benefits to humanity. It can provide opportunities to accurately diagnose, prevent, or cure a wide range of infectious and genetic diseases. Significantly increase crop yields by creating plants that are resistant to insect predation, fungal and viral diseases, and environmental stresses such as short-term drought and excessive heat, and at the same time reduce applications of hazardous agrochemicals. Develop microorganisms that will produce chemicals, antibiotics, polymers, amino acids, enzymes, and various food additives that are important for food production and other industries. Develop livestock and other animals that have genetically enhanced attributes. Facilitate the removal of pollutants and waste materials from the environment. Although it is exciting and important to emphasize the positive aspects of new advances, there are also social concerns and consequences that must be addressed. The following are some examples. Will some genetically engineered organisms be harmful either to other organisms or to the environment? Will the development and use of genetically engineered organisms reduce natural genetic diversity? Should humans be genetically engineered? Will new diagnostic procedures undermine individual privacy? Will financial support for molecular biotechnology constrain the development of other important technologies? Will the emphasis on commercial success mean that the benefits of molecular biotechnology will be available only to wealthier nations? Will agriculture and molecular biotechnology undermine traditional farming practices? While and one among the figures shown in this, there's it's an artist's impression, so it seems a bit graphic, so I think explaining that would be quite difficult for newcomers. So we'll have another reference. I'll just give you a general idea of what the thing is about. There's a farm that explores iconography of agriculture, A disproportionately enormous and savage cow has an overabundance of teats. The pig is a human organ factory, and the chicken, which boasts 3 pairs of wings and no feathers, is ready for basting. The fruit fly, the workhorse of many genetic study, is present as is a mouse with a human ear cartilage projecting from its back. Past, present, and future states are threaded together here with the barbed wire, woven baskets, and DNA. So just about the upcoming and the trends and the obstructions, hindrances, and promotions all together. The farm shows how the bodies of these animals have been and may one day be transformed to suit our aesthetic, medical, and gastronomic needs. And will medical therapies based on molecular biotechnology supersede equally effective traditional treatments? Will the quest for patents inhibit the free exchange of ideas among research scientists? These and many other issues have been considered by government commissions, discussed extensively at conferences, and thoughtfully debated and analyzed by individuals in both popular and academic publications. On these bases, rules and regulations have been formulated, guidelines have been established, and policies have been created. There have been active and extensive participation by both scientists and the general public in deciding how molecular biotechnology should proceed, although some controversies still remain. Molecular biotechnology, which with much fuss and fanfare became a comprehensive scientific and commercial venture in a remarkably short time. Many scientific and business publications are now devoted to the subject, and graduate and undergraduate programs and courses are available at universities throughout the world to teach it. When artists have depicted their perception of molecular biotechnology, it could be debated whether the early promise of biotechnology has been fulfilled in the way that was predicted in a 1987 document published by the U.S. Office of Technology Assessment, which declared that molecular biotechnology is a new scientific revolution that could change the lives and futures of citizens as dramatically as did the Industrial Revolution 2 centuries ago and computer revolution today. The ability to manipulate genetic material to achieve specific outcomes in living organisms promises major changes in many aspects of modern life. It does, however, offer solutions to some serious global problems, including the spread of infectious diseases, the burden of waste accumulation, and food shortages. The potential of molecular biotechnology to solve some of these imminent problems is the subject of this book. And for a summary, well, before I could read the summary, there's another note that I need to read before you. Just a second while I just fetch that for you. Yeah, this was the one. Now, this is about a landmark study of KORAH-NET established the foundations for about KORAH-NET, and they've established study on that for recombinant DNA technology by showing how genetic information, from different sources could be joined to create a novel replicatable genetic structure. In this instance, the new genetic entities were derived from bacterial autonomously replicating extrachromosomal DNA structures called plasmids. In a previous study, Cohen and Chang produced a small plasmid from a naturally occurring plasmid by shearing the larger plasmid into small random pieces and introducing the mixture of pieces into a host cell, the bacterium E. coli. By chance, one of the fragments that was about 1/10 the size of the original plasmid was perpetuated as a functional plasmid. And to overcome the randomness of this approach and to make the genetic approach, the manipulation of plasmids more manageable, Cohen and his co-workers decided to use an enzyme restriction endonuclease that cuts a DNA molecule at a specific site and produces a short extension at each end. The extensions of the cut ends of a restriction endonuclease treat a DNA molecule and can combine with the extensions of another DNA molecule that has been cleaved with the same restriction endonuclease. Consequently, when DNA molecules from different sources are treated with the same restriction endonuclease and mixed together, new DNA combinations that never existed before can be formed. In this way, Cohen et al. Not only introduced a gene from one plasmid into another plasmid, but also demonstrated that the introduced gene was biologically active. To their credit, these authors fully appreciated that their strategy was potentially useful for insertion of specific sequences from prokaryotic or eukaryotic chromosomes or extrachromosomal DNA into independently replicating bacterial plasmids. In other words, any gene for any organism could theoretically be cloned into a plasmid, which after introduction into a host cell would be maintained indefinitely and perhaps produce the perfect protein encoded by the cloned gene. By demonstrating the feasibility of gene coding, Cohen et al. Provided the experimental basis for recombinant DNA technology, established that plasmids could act as vehicles vectors for maintaining cloned genes, motivated others to pursue research in this area to rapidly led to the development of more sophisticated vectors and gene cloning strategies. Endangered concerns about the safety and ethics of this kind of research that, in turn, was responsible for the re-establishment of official guidelines and government agencies for conducting the regulating recombinant DNA research, respectively, and contributed to the formation of the molecular biotechnology industry. Right. And now for a summary on today's lesson. In 1973, Stanley Cohen, Herbert Boyer, and their co-workers devised a method for transferring genetic information, genes, from one organism to another. This procedure, which became known as recombinant DNA technology, enabled researchers to isolate specific genes and to perpetuate them in host organisms. Recombinant DNA technology has been beneficial to many different areas of study. However, its impact on biotechnology has been extraordinary. Biotechnology, for the most part, uses microorganisms on a large scale for the production of commercially important products. Before the advent of recombinant DNA technology, the most effective way of increasing the productivity of an organism was to induce mutations and then use selection procedures to identify organisms with the superior traits. This process was not foolproof. It was time-consuming, labor-intensive, and costly. Only a small set of traits could be enhanced. In this way, recombinant DNA technology, however, provided a rapid, efficient, and powerful means for creating microorganisms with specific genetic attributes. Moreover, the tools of recombinant DNA technology enable not only microorganisms, but also plants and animals to be genetically engineered. Combining recombinant DNA technology with biotechnology created a dynamic and exciting discipline called molecular biotechnology. From its beginning, molecular biotechnology captured the imagination of the public. Many small companies dedicated to gene cloning, recombinant DNA technology were established with funding from private investors. Although these biotechnology companies took somewhat longer than expected to bring their products to the marketplace, a large number of recombinant DNA-based products are currently available, and many more expected soon. Because of its broad impact, molecular biotechnology has been scrutinized carefully for its potential effects on society. Some of the concerns that have been raised are its safety, its possible negative effects on the environment, and the private or public ownership of genetically engineered organisms. We have the Q&A to sum up, we'll have the Q&A for this session. Right, so that would be questions on the completed chapter 1. So what is biotechnology? These are review questions. What is biotechnology? Distinguish between traditional biotechnology and molecular biotechnology. Describe the basic steps of a bioengineered technology process. What are the basic steps of a biotechnology process? What are the shortcomings of the mutation and selection method for developing enhanced organisms for commercial purposes? What was the work reported by Cohen and Boyer and the co-workers of 73 and 72 considered important? How did recombinant DNA technology enable the production of human insulin? What are some of the problems that molecular biotechnology has the potential to solve? Discuss the statement molecular biotechnology is a diverse science. Discuss some of the social concerns that have been raised about biotechnology. That should be it. So I'll see you in the next lecture a couple of minutes from now. Take notes, prepare notes, and study well. Best wishes.